Well, thank you very much, Jean-Jacques, for inviting me here. I'm very pleased to be here. I've only been here once before at this very august institution. So what I then wanted to do over the next 50 minutes or so is to talk to you about our efforts to retrieve the genomes, the genetic material from our closest extinct relatives. You hear me in the back? You'll have to tell me if it doesn't work. And I would then just like, towards the end, more talk about what we have learned about studying these genomes and what we might learn in the future by using these genomes. But I wanted to just start out by reminding you all about the fact that you already know, that our genetic material is stored in all cells in our body, almost all cells in our body, and that the information is stored sort of twice, once on each strand in the sequence of these 4 nucleotides abbreviated A, T, C, and G. And as you also know, it's really important that information is there twice, because whenever the genome divides, when a new cell should emerge, these strands come apart and new ones are synthesized with the old as a template. This process is very exact, but sometimes, of course, errors are made. Sometimes an incorrect nucleotide or base is built in. And if that is not repaired quickly enough, it will be inherited to the daughter cell as a mutation. And if that happens in a germline, it will be a mutation that appears in the new individual that is born. So every baby that's born carries somewhere in the order of 100 or 200 new mutations that is not there in the mother nor in the father. And these mutations, the traces of them or the results of them, we can see if we compare a DNA sequence in the population. So if we compare 2 genomes in this room, we find a difference between 2 individuals approximately once every 1,000 nucleotides. And since these mutations rain down on our genome in each generation, if we then go on and compare ourselves to a chimpanzee, we will find more such differences, about 1 every 100 nucleotides. And if we're then interested in the evolution, the history of a piece of DNA or a genome, we use these differences to reconstruct the history of the genomes. And in this case, it's really simple. These 2 human genomes go back to a common ancestor rather recently, have about 1 difference every 1,000 nucleotides. If we go to the common ancestor with the chimpanzee, we have about 10 times more differences, so we're much further back in the time. And you also know that our genome is about 3 billion such nucleotides. So we have something in the order of 3 million differences between 2 genomes. So there is a lot of variation there to help us reconstruct our genomic history. And if we now do that on a worldwide scale, what one then finds is that we have most genetic variation in Africa. And if we take the entire world outside Africa, although there are a lot more people, of course, who live outside Africa than inside Africa, we have less genetic variation there. And not only that, the genetic variants we find outside Africa generally have closely related variants inside Africa. But there is a proportion of the variation in Africa then that has no close relatives outside Africa. So the interpretation from that pattern is then that modern humans, the direct ancestors of present-day people, evolved in Africa, accumulated genetic variation there, and a part of that variation, so to say, went out and colonized the rest of the world. And with some genetic tricks, for example, by seeing over how long distances on chromosomes you have these sort of patterns